Thank you so much for being here. Welcome to our seminar series, Cutting Edge Advances Towards Personalized Mental Health, hosted by the Center for Precision Mental Health and Wellness. My name is Divya Rajasekaran, and I'm a PhD student in the center advised by Dr. Leanne Williams. I have the privilege of working on our neuroimaging TMS study in partnership with the VA clinical TMS program. So I'm thrilled to have Dr. Jonathan Downar join us today to discuss the current landscape of clinical TMS and opportunities to scale access to treatment. Before we get started, I'm going to share a few housekeeping notes. So one, the webinar is being recorded and we will post it on our website next week. You can find our website in the chat if you're on Zoom, or scan the QR code on the left side of the screen. From there, you can also sign up for announcements for our future events. If you're interested in joining our center as a research member, please see the link in the chat or scan the QR code on the right side of the screen to fill out a survey. And lastly, if you're on Zoom, please put your questions in the Q&A box and we'll attend to them at the end of the presentation. Feel free to post your questions as early as possible since we're going to address them in that order. And if you're here in person, you'll be able to raise your hand to ask our speaker questions directly at the end of the presentation. And with that, I'll hand things over to the director of our center, Dr. Leanne Williams, who will introduce our seminar speaker today, Dr. Jonathan Downar.
Thank you so much, Divya. Divya is doing incredible work on our TMS project, and I'm sure we'll have that presented in the near future. So today, it's an absolute privilege to welcome you, Jonathan Downar, as today's keynote speaker in our seminar series. As many of you know, Dr. Downar is an adjunct professor in the Department of Psychiatry and the Institute of Medical Science at the University of Toronto, and also holds a clinical appointment at the Toronto Centre for Addiction and Mental Health. He completed his medical training at the University of Calgary, and his PhD specializing in functional neuroimaging and psychiatry residency were completed at the University of Toronto. We have been very inspired by Dr. Downar's work. He's a leader in our field. He's published over 180 research articles in impactful journals that include The Lancet, Nature Medicine, Nature Neuroscience, JAMA Psychiatry, and the American Journal of Psychiatry. His groundbreaking 3D study was published in Lancet in 2018, and it revolutionized TMS by demonstrating that sessions could be reduced from 38 minutes to just 3 minutes without compromising efficiency. He's also pioneered novel TMS protocols, including with the dorsomedial and orbitofrontal regions as targets, expanding applications in depression and other disorders. As we'll hear today, continuing to push the boundaries, Dr. Downar is developing a new generation of accelerated TMS protocols incorporating pharmacological enhancements and scalable technology to expand both access and impact. Please join me in warmly welcoming Dr. Jonathan Downar, who will present on 1 billion remissions in our lifetime, how we might achieve universal access to non-invasive brain stimulation therapies.
Lee, thank you so much for such a warm introduction, and Divya as well. Thank you. It's delightful to be invited here and delightful to see all the progress and change that have happened since last time I was here a number of years back. So thanks for having me, and look forward to chatting with you. We are talking about this. Obviously, everyone here has been working very hard to understand the mechanisms of TMS and to understand how to personalize it and to understand how to get patients better. And the more, having put decades into all that work, we'll all feel a whole lot better the more patients that can reach over time. So, the more remissions we can achieve and the further out we can scale all of this, the more impactful our work's going to be. And so while all the hard work goes into the brain imaging and the brain mapping and the stimulation and so on, I've been keeping one eye on scalability just to make sure that all the hard work that all of us are doing does try to get out into the real world to reach as many people as possible. There is a crazy, what I'm going to call a loon shot to reach 1 billion people in our lifetime. Is this a completely ridiculous idea that we could use brain stimulation to improve the lives of 1 billion people? I have a unit of global impact, which I call the Borlaug, after Norman Borlaug, who was, we believe, the most impactful human who ever lived. He was the essentially the creator of the Green Revolution back in the 1960s. And being a humble crop breeder initially, he just figured out ways to sort of fiddle with grain and fiddle with its resistance to fungus and cause it to yield more per acre. And his agricultural practices spread around the world, and pretty much universally credited with having saved 1 billion people from starvation over time. And the lovely part is no one's ever heard of him. So the Borlaug, Norman Borlaug, the most impactful human who ever lived. And so we can create a metric unit called the Borlaug, which is 1 billion 1 billion people positively impacted. I believe about 100,000 people a year are now getting treated with TMS using the theta burst protocol, the 3-unit protocol we talked about. So that's about 100 microborlogs of impact a year. And so we'd like to get up to the milliborlogs and maybe even up to a full borlog one day. So that's the goal here. Is this possible to do? And is it possible to do with TMS? Well, TMS for depression is now comfortably middle-aged. If any of you are under 30 here, TMS for depression is now older than you are. So, the idea was that in the 1980s, there were PET scans showing what areas of the brain were underactive in depression. The left dorsolateral prefrontal cortex helpfully pointed out with an arrow, and maybe we should put a coil here and see what that does. And it turned out in 1995 that the first demonstrations that did indeed improve depression were helpful. So if we say that around by the year 2000, we had maybe 1,000 people a year getting TMS for depression, if you go across all the studies and so on. Interestingly, a 20% growth rate, which is about what TMS is growing at, has been fairly consistent from the year 2000 over the last 25 years to year 2025. There are now about 120,000 people getting TMS in the US every year. So there's been a very respectable 20% growth rate. And if we do nothing about that whatsoever, then the miracle of compound interest will still get us to about 10 million people a year being treated with TMS by the year 2050. But if we want that billion remissions a year, then we really need to bump it up, not ridiculously. Getting it to 40% will just about do it. Getting it to 60% growth rate a year will absolutely do it by the mid 2040s. So it's not a completely ridiculous idea. We simply just have to figure out, is there some way to boost the remission rate a little bit from 20% to 30 or 40%? And that means addressing the great logistical barriers to TMS adoption. The two things that everybody complains about TMS, no matter how good it gets, which is number one, it's too expensive. $10,000 per patient is the going rate, and a lot of prices higher in some places, depending on how wealthy your neighborhood is. It's not universally scalable in the way medications are. So the first crazy idea is cost parity of TMS with medications physically or logically possible? Because if so, it might be scalable. If not, then not. And then the second part is convenience. Number of times I've probably personally chatted with maybe 5,000 or 6,000 patients about TMS. A lot of them say, I just can't come in for 36 visits. It's just not doable. Fortunately, right here at Stanford, our good friend Nolan Williams was the developer of a 5-day protocol. Amazing, the SAINT protocol. And you still get people who say that's still not good enough. I still can't come for a whole week. It's really frustrating. And then they say things like, well, I typed this into deep research on ChatGPT and it says I should do psilocybin because that's a 1-day treatment. So can I have 1 day? What does 1-day TMS do? Could I just do 1 day? Patients ask the best questions, right? And so the question becomes, could I do 1 day of TMS? And I realized I didn't know what the answer was. I didn't know whether to tell them it's a waste of time, the remission rate's been studied, it's been 5%, or it's a great idea, the remission rate's like 80%. We have no idea. So I have to start thinking about, well, what's the maximum you could do for somebody in a day? And to get into that, we need to do a bit of a deep dive into the mechanisms of TMS to address these. Number one, what we always think about with mechanisms is the neuroanatomy. What are the targets for TMS and depression? And of course, as far back as 1989, we already had little PET scans showing us where we could put coils that might be helpful in depression. You notice that there's, the left dorsolateral cortex, prefrontal cortex with the arrow is the one everybody loves, but there's lots of other lovely targets here that are on the map that don't have arrows on them, but should. And as you mentioned, the dorsal medial's there, and there are even some lovely parietal areas that are being investigated over at Harvard and so on. And there are also some deep areas you can't get to superficially. These are areas like the ones that Helen Mayberg targeted back in 2005 with her subgenual stimulation. And what's lovely is when you implant electrodes in the subgenual areas, not only do you turn off the subgenual cingulate cortex, but what turns on are, in red here, some of these very same areas that you see as being underactive in those original PET scans. So a nice correspondence there that, more than a decade ago, was spotted as being potentially relevant by Mike Fox and colleagues and saying, oh, there's a relationship here. We've got this subgenual target that works for DBS, and it's anti-correlated to a bunch of regions in resting state functional connectivity. And if we really want TMS to shine, what we need to do is stop using tape measures and figure out where this spot is in each person and get the coil on there. Because across various trials I looked at, this is all the way back in 2012, there is a sort of optimal spot, and the further away you are from it in these trials, the less well people did. Lovely idea. We adopted a generic target from this for the 3D study to try and improve outcomes. We didn't get 100% remission or anything like that because it was a generic target. We did not personalize the target for each person. But the question becomes, how close do you have to be within the millimeter? Do you have to be within 2 millimeters? Because even when you use state-of-the-art functional imaging with stereotactic frameless navigation and so on, there's a 3 millimeter calibration error just on the coil. There's another 3 millimeter calibration error on the head. So we've had people do this over and over again. And here, I find our best techs, if you use invisible ink pens so they can't cheat, and you just have them do it over and over again, you still get a scatter of about 8 millimeters or so just from session to session. But that does seem to be kind of good enough. Robin Cash and his colleagues a few years back started looking at least in a smallish sample, that it's very hard to get more than about 20% improvement in the MADRS if you're not within at least 25 millimeters or so. So that's not terrible. And 25 millimeters is pretty fuzzy. And ideally, we want to be even closer than that. But I don't actually want to delve too deeply into how close you have to be on the target. What I want to ask is the counter argument, which I think people haven't been getting into much, which is it possible to be on target and still have nothing happen, right? How often could you be exactly on the right target? And is it ever possible that something, and the problem is that the answer looks like yes. Charlotte Neckhoven's paper, which should be quoted a lot more than it is, from 10 years ago now, she was working in motor cortex, which is a very handy spot because you know whether you're on target or not because the thumb moves. Again, the SAINT protocol draws upon some of this evidence in support of using the 1800 pulses, noting that when you do 600 pulses, you get a boost. You do another 600 pulses, 15 minutes later, no boost, and then you get another 600 pulses, and you get another boost in the motor evoked potential. And then here's the sham group at the bottom showing that sham stimulation doesn't do that. The problem is, if you read the fine print, this was 7 out of 16 of the subjects in the study. All the others were non-responders, motor cortex non-responders, where the first 600 pulses did nothing, this next 600 did nothing, and the next 600 also did nothing. Ouch. And that's right on target. The thumb is moving, so you know you can't be off target. And so annoyingly, it looks like some people just don't have plasticity. We think we see this. So even in the 3D study, we look at the true number of non-respond. We find that there's a mean of how well people improve, but if you break it down into subcategories, there seem to be 4 main trajectories of response to TMS, at least in our 3D and some other studies. There are fast responders, and those of you who practice TMS see those amazing people who are fully better in 18 or 20 sessions, but it's only like 1 out of 5 people, and that's what we saw in theta. But then, interestingly, the true non-responders who were completely flat and got nothing were 10 to 12% of the sample. We were blown away by this, that really only 12% of people show nothing. And everyone else is in the middle. They're kind of we're bailing these leaky boats where we're kind of putting a few pulses in and then it's kind of muddling along. And we just wish they had as much plasticity as the fast responders. So the question would become, there's probably another part to the mechanism of TMS, because with the deep brain simulator, once you're in the right neuroanatomy, that thing's stuck in your head all day and you're doing pulses and the person is not going to, with TMS, you have to do more than that. You need plasticity. Because they're not walking around with the stimulator in their head all day. So you're going to do 3 minutes of pulses or 10 minutes of pulses or whatever, but it's got to last for weeks. Otherwise, it's just not going to pile up. So you need plasticity, and that means we need to also delve into the part 2, the neglected part of the mechanism of TMS, which is no plasticity, no TMS effect. How do we enhance neuroplasticity? The literature provides 2 main ways forward. Preclinically, dopamine. Dopamine is really, really helpful, and not just any dopamine. If you apply pramipexole, which is a dopamine D234 agonist, it does nothing. It does not enhance plasticity at all. If you use L-DOPA, which is metabolized to dopamine itself and hitting the D1 and the D2 receptor, then what's interesting is very inhibitory protocols like water pulse and so on become more inhibitory, and very excitatory protocols also become more excitatory. So they are not so much a up or down, they are an enhancer of whatever direction the plasticity is going in. The other finding was from clinical work out of UCLA by Scott Wilkie from Andy Luchter's group there. Having dug through all of their records, they found that patients who happen to, so by the way, the implication of this is, dopamine should boost things up, but you don't find that many patients on L-DOPA doing TMS. You do find a lot of patients on stimulants doing TMS, and that also puts a lot of dopamine at the synapse. And lo and behold, patients on dextroamphetamine or lisdexamfetamine or Concerta or methylphenidate, any of these, they all seem to have better outcomes, faster responses. And then modafinil, which is a non-dopaminergic agent, does not do anything. So nice little sort of study there. So dopaminergic augmentation could be helpful. The other big one, which I think is getting a lot of attention right now, is the NMDA pathway. D-cycloserine, the NMDA agonist, Josh Brown and their group over at Harvard have shown that there's some enhancement preclinically of the motor evoked potentials using these partial agonists at NMDA. But the real kicker was Alex McGeer and colleagues over at University of Calgary back in 2022. Even with a very stingy Canadian course of 20 sessions, you can see quite a lot of effect here. So 20 sessions is not expected to do that much. Among clinical practitioners, the rule of thumb is you get 1% remission for every one session you do. So the early studies that did only 15 or 20 sessions of stimulation back in the day were responsible for this premise that TMS only has a 20% remission rate. You do 30 sessions, you get a 30% remission rate. You do 50 sessions, you get and it doesn't go on forever, but as a rough rule of thumb. In this case, the results kind of fit with that. If you took people and gave them 20 days of TMS using theta burst stimulation over F3, no personalized target, just over F3, you get about an 18.9% remission rate. So it's kind of what you expect. But if they're taking D-cycloserine, even just for the first 10 of those days out of 20, the remission rate is now 40% and the response rate is 70%. It's a huge jump. Like nothing does that. And then, Alex has had some lovely communication. You've seen he's just hot another paper out showing an OCD that, off D-cycloserine, the Y-box improvement on 20 sessions is about 2 points, and it's closer to 10 points when you're on there. So, and that's with dorsomedial, and that's for a different indication. Very, very useful information. Now, let's come back to our patient's question. So if a patient comes in and says, well, my psilocybin clinic says they can treat me in a day, and so I want to come to your TMS clinic and I want to get 1 day and see what that happens. Well, we'd be a little bit foolish not to try to incorporate some of this to just give people the biggest bang for the buck. And the question is what's the shortest course of TMS anyone's ever done? Well, back in 2010, Paul Holzheimer gave people 1.5 days of TMS, 15 sessions. And this is old-fashioned sort of 10 hertz TMS, totally unoptimized, 1 session per hour, kind of a proto-saint for a day and a half, and a very respectable, like 30-ish% remission rate, which was held out to 6 weeks. And after this groundbreaking, open label trial in 2010, what happened after that was exactly nothing. No one ever replicated it. It was really weird. I remember I was just opening my TMS clinic in 2010 in Toronto when this paper came out. Oh my goodness, like maybe we can do TMS in a day or two. And we all just got busy with other things. And for a long time, nobody checked it until Nolan popped up. And well, other people looked at Accelerated, but nobody else came back to do 10 sessions a day until Nolan had a crack at it. And obviously that worked out pretty well. But what does 1 day of sync do, right? Or, if that's it, or even let's say we're in a clinic where we don't have access to personalized neuroimaging targeting, but we do have D-cycloserine, we do have dopamine, we have things like that. So we could put together a thing which I'm not calling 3D, but 1D, an optimized neuromodulation enhanced with D-cycloserine and dextroamphetamine. So fine, if you are saying I'm totally not willing to come in for 36 days, I can't even find 5 days, but I can come in on Saturday as long as you can get everything done on Saturday, because I live 4 hours away. So I've got 1 day, I can come down Saturday, get a bunch of sessions. What's the best you can do for me? Well, let's make sure that we use D-cycloserine, because that seems to be really helpful in boosting remission rates. Let's see if we can squeeze the interval from 60 minutes down to 30 minutes, because, Charlotte Netkoven, again, back in that paper, the first session gave a boost. The second session, 15 minutes later, didn't do anything. But the third session, 30 minutes later, did give another boost. So let's just see if we can, and also Andre Bernoni has recently published another trial using a 30-minute interval for a kind of 3-session a day. So if I use a 30-minute interval, I can squeeze in 20 sessions in the day. And that's nice because 20 sessions on D-cycloserine seems to do quite a lot. Let's also make sure we augment dopamine. So I'm not going to give people L-Dopa, but in Canada, Vyvanse is pretty plentiful, and so we're going to use Vyvanse there, light dose, 20 milligrams, and a 1-day protocol. So we're going to squeeze this all into a single day. It's a very intense day. The people show up at 6:30 a.m. and they take right on the spot. They take a compounded dose of 125 milligrams of rapid dissolving D-cycloserine made by a compounding pharmacy. They also take 20 milligrams, light dose, 20 milligrams of lisdexamfetamine. And then you've got 1 hour for it to kick in. So during that hour, you do the motor threshold and you collect all their baseline scales and their Hamilton, and you get them all ready. And then every 30 minutes, they sit in the chair and you get 3 minutes of theta bursts. Not a personalized target. Just, I'm showing you, it's actually Sean Siddiqui's lesion-based posterior target, partly because we like the fact that it's lesion-based and causally network mapped, but also it's further back than BMF3, so it's easier to tolerate for these poor patients who are having to do 20 sessions, totally naive in a day. So all of that happened. What kind of patients? Well, these are representative patients from community clinics. These are not sort of strange, weird patients. These are just patients who showed up, and for every once in a while, I understand that sometimes insurance turns down TMS for spurious reasons. I'm told that happens here sometimes. So these patients who are just, for whatever reason, not accepted by their insurance, or they said, I'm accepted, but I just can't come for 36 days. But in every other respect, adults 18 to 65, average have failed 3 antidepressant trials. They all had to have a PHQ-9 over 15, or at least the ones we're including here did. And standard outcome measures. This is all off-label prescribing, so this is all is kind of an off-label. This isn't the usual way we do TMS, but if you're willing to do off-label and this is the only way you'll do it, then let's offer it to you. No guarantees as to what will happen. What does happen is the throughput is amazing. Typically, on most protocols, you can only treat 2 or 3 patients per week on a given machine. Just like on average, you get a throughput of 2 or 3 patients per week on a machine. Here, they're doing 3 patients a day because you're doing 3 minutes. You bring 3 patients in. One of them goes at 9 o'clock, one of them goes at 9:10, one goes at 9:20, and then you just start again. So this bizarre world where people you can run 15 patients through in a week. So trials can be done very quickly as long as you have enough patients. It's one of the interesting, and I think there are research implications from that as well if you want to test out hypotheses. All of which means nothing. The success rate is terrible. So I didn't know what was going to happen. I did know that we should probably not expect people to get better right away because it's Saint. Anyone who practices accelerated knows they're often not all the way better right away. It's not ketamine, and sometimes there's a delayed effect. And so we decided to measure people weekly out to 6 weeks and see what happened. I didn't expect this to happen at all. What happens is you get this lovely exponential curve where people show some improvement on week 1, more improvement on week 2, more improvement. It takes about 6 weeks for maximum improvement. But because you enhance all this plasticity, the durability is pretty wild. Like they're actually showing still great effects out to 12 weeks, so 3 months out. We're now getting our 6-week data back, and some are relapsing, but I'd say about 80% of them are still doing really well 6 months later off a single-day trial. And this was the other part that was ridiculous. I told you earlier that only 10% of people are true non-responders in their 3D trajectory. I didn't expect that all of the medium responders would somehow get flipped over into being something. We actually got only about 10% of people here being complete non-responders, but 90% of people showed at least a 50% improvement, which we had no idea this would happen. What I thought would happen is we do a 1-day treatment. It would help maybe 30% of people. Then we go back and have to do it again for a 2-day and then a 3-day. And then we just kind of gradually build a curve. I did not expect that most of them would get better on the 1-day version. But what's interesting is you'll also notice the complete lack of correlation between the intervention timeline and the outcome timeline. So we have always told ourselves that the magnetic pulses are only doing a bit of the work of TMS, that a lot of the work of TMS is coming in every day, having a routine, behavioral activation, social interaction, having a nice tech who's sort of doing mini therapy with you while you're getting but these people don't get any of that. It's one day, and then you're back home to your regular life. And the day later, nothing, very little happened. Very few people walk in the next day saying they're doing better. It develops over weeks, and we can unpack as to why that might be over time. But it seems to be very consistent across 4 different scales: the Hamilton, the PHQ-9, the Beck Depression Inventory, and the GAD-7. Each one with a trace here showed very consistent. Oh, that was interesting. The GAD-7, by the way, anxiety effect size was just as big as all the others with this target. We didn't think anyone would believe the numbers. We were looking for ways to make the numbers worse for an abstract we were submitting. And so we decided we would only call them, to be a remitter, they had to remit on all 4 of the scales. And to be a responder, they had to remit on all 4 of the scales. And even with these like strict criteria, we still had 84% of people meeting response and more than 50% of people meeting remission, even if you say you're only a remitter if you remit on all 4 scales. So doesn't take too many sessions. The thing is, it's a ridiculous claim, but at the same time, it's also very easy to falsify. So I thought people would call me up and say it doesn't make any sense. And what they did, they called me up and they said, I don't believe you. So I tried it, and then it's yeah, it does seem to work. And so it will speed up research quite a lot if we get this going. So this is what's in the 1D preprint, and the recipe is out there. And while it's under review, there are people out there who are calling in and telling me they've tried it out to see what's going on. We have gone back to try this again in people with an even more prevalent disorder of generalized anxiety. And so this is new data I haven't shown yet. We're only out to about 5 weeks with these folks, and it's a smaller sample so far, about 17 people. But these folks presented not with depression, but with generalized anxiety. And they came in, we just gave them exactly the same protocol. The only difference is no Vyvanse. People have objected to the Vyvanse and said, well, I don't like this idea of putting people on Vyvanse. And you guys in Canada can do what you want, but in the US, it's a controlled substance and so on and so on. So, can we just do it without the Vyvanse? These folks, being anxious, we elected to omit the psychostimulant from their regimen, and they still did pretty well. We're still seeing quite a lot of remitters and responders at 5 weeks on the Hamilton Anxiety Scale and the GAD-7. If you plot this in terms of percent improvement over time, it's looking pretty good. Comparable benefit at 60 to 70% improvement at 5 weeks across the 4 scales. 1D was showing more like a 75 to 85% improvement at 5 weeks. There's a lot of non-comparables between these. You have 2 different kinds of populations. They're not exactly the same sample. It's not a randomized comparison, yada, yada. So we probably will need a randomized control trial to figure this out, but they're kind of ballpark the same. It looks like adding the Vyvanse might do a little bit more, but we're not really going to know for sure until we sit down and do this as a proper randomized control trial, which I think is something to do as soon as we finish optimizing the protocol. And there are broadenings beyond anxiety as well. So we also had a couple of folks show up with OCD. As I mentioned, Alex McGeer did a lovely study, which has just been out with a commentary by Josh Brown and Noah Phillip as well, talking about the use of this in OCD. So we did have a couple of people coming in for 1 day of treatment on OCD. The first one came in with a Y box of 26 at baseline, walked in the next week with a 7, and she's still down there 6 months later. That's kind of nuts. And this is like a true OCD person. She'd been coming to the clinic for years, not really responding to anything. And, the typical kind of 2 decades of contamination and various other fears. She's still doing great. She was OCD with comorbid depression, as you can see by the Beck scores and the PHQ-9 scores there. Connor Lisson and Nolan Williams have been doing some really lovely work biotyping and doing a Saint-style protocol in OCD. And Connor seems to have arrived at at least 2 subsets, one of which is characterized by having OCD plus depression, and the other is characterized by having OCD without anything else other than just the OCD. The ones with the comorbidities seem to do well on dorsomedial in his analysis, and the others do better on orbitofrontal. This was dorsomedial. We had another case of a person who came in without the comorbidities. Here's another OCD who just had OCD, no depression whatsoever. Still not bad, 30-35% improvement, but makes me wonder if we should go back and give this person orbitofrontal and try again, because this was probably not the right target for theirs. But the point is, it's still, what difference does a day make in an OCD patient? You can get quite a lot of traction in 1 day, at least in some people. So this is another population to be kind of broken out a bit. All right, back to the loon shot. Not the moonshot, the loon shot. 1 billion remissions in our lifetime. Let's forget about whether it's possible. Let's forget about whether it sounds crazy or not, and just ask the question of what would it have to be true? What would it take to get there? And probably also, like, what do you mean remissions from what exactly, is probably a question that we'll get to in a second. But there are, when we say moonshot, let's go to actual moonshots. Let's go to actually getting stuff to the moon and how much that costs and how doable that is. We wanted to, I don't know, build a city on the moon or something like that. Here, you need to start looking at economics. You need to start looking at metrics like how much does it cost to launch a kilogram of stuff up into orbit. And in the very early days of the space race back in the 1960s, it used to cost almost almost $1 million per kilogram to get something up into orbit. And over the course of the next few years, by the time they got to the Saturn V, it was down to a lot less than that. And once you have the Saturn V, it was possible with the resources of a large nation like the United States to launch some stuff up into orbit and at least land a spacecraft on the moon, go around and play golf and collect some rocks and then fly back again. That was doable. Not possible to send millions of tons of gear up there and build an entire city. It's just the economics just don't work. And then we had all the lost decades. Annoyingly, right from the time I was born up until fairly recently. I don't know how many. I thought we were all going to be living in cities on the moon by now. And I was like, for those of us who were of this age, we all thought there'd be all kinds of stuff up there. Why didn't it happen? Well, one hint is the economics of space flight didn't change for decades, right? It didn't. The cost of flying stuff into orbit just didn't come down. And it was only in the SpaceX era that somebody focused specifically on things like reusable rockets and how do we get the cost of stuff to orbit down. And they just focused relentlessly on that metric. We will redesign the rockets, we'll land them on barges, we'll do all kinds of crazy stuff, we'll reuse them, whatever it takes to get the cost per kilogram down as low as possible. We still don't have cities on Mars or the moon yet, but we do have, thousands of Starlink satellites in orbit providing internet all over the place. And that just would not have been possible with the economics of spaceflight 10 years ago even. So, and now, of course, if you want to build a city on Mars, you have to get even lower than this, right? You actually have to get it down to like under 100 kilograms. And that's where you have to do crazy stuff like catch the rocket back on its launching pad. And you've all seen SpaceX do these ridiculous things of trying to fly rockets all the way up and land them again. The idea is just to reduce the cost of launching to just the fuel and just fly them up there, launch the stuff, fly back again and refuel. So it just becomes the cost of the fuel, about $2 million of fuel per launch, which means about $20 per kilogram of stuff to orbit. And now it's actually possible to send 1 million tons of gear up to Mars, up to the moon or Mars and build a city. So what is the corresponding metric for TMS? I'm going to call it cost of delivery. Like how much does it cost to do TMS? And rather than break down the device and the technician and all the rest of it, interestingly, the cost of a TMS device by the hour is maybe only $3 to $5 an hour. So if you actually rented a TMS device by the hour, the technician's another 25. Then there's a lot of overhead and administration. Typically, a staffed exam room in a family medicine clinic or whatever falls around $80 to $100 per hour. So we'll conservatively say that your cost of delivery, if you have a technician, you have a TMS machine, and all the rest of your cost of actually delivering this stuff is, we'll call it ballpark $100 an hour. And now let's go back and look at the total room time. Room time is how long are your sessions, and then how long does it take to get somebody out of the chair, and then how many sessions are you doing? The original 10 hertz O'Riordan protocol that was FDA approved, 36 sessions of 38 minutes duration, you're talking almost 22.5 hours of brain stimulation alone. And with the added transition time of 8 minutes to get people in and out of that, and that's generous. Some places take longer. No wonder TMS the cost of delivering it alone is almost $3,000 just because that's how much, it would be like a surgical procedure that needed 24 hours of OR time. You're just never going to be able to do that. And then, it was many years before the DASH protocol came along that cut at least the brain stimulation time from 38 minutes down to 19 minutes. And Brainsway developed their own protocol of 20 hertz, which is a 20-minute protocol. And then Brainsway chairs are a bit faster to get people in and out of because placing the helmet is a little bit speedier than placing a coil. So you get some advantages there. Saint offers these great remission rates, but because you're doing 50 sessions and because you're per session, you actually still end up the total cost of delivery. It's a lot better than O'Riordan was, about half of that, but you're still got almost 1,000 minutes of room time per patient to do Saint. 3D doesn't offer the remission rate of Saint, but 3D, one thing about 3D, it's only 3 minutes of treatment. And even with the transition times, you are only looking at consuming about 400 and something minutes. And so the cost of delivering 3D is actually really low. And this is a classic Canadian thing where Canada just doesn't want to spend very much money on stuff. And so we needed an inexpensive treatment. One thing that doesn't jump out obviously about the 1D protocol is that it's 3 minutes times 20 sessions. That's 1 hour. You're doing the entire treatment with this, whatever remission rates, in 1 hour of chair time. And then room time, the transition time of 8 minutes, you actually are even faster getting people in and out of the chair when they're just there all day and they're not coming and going and coming and going. I've even had a couple of clinics out there which have reduced, instead of using the 3-minute treatments, they're using CTBS. CTBS is 33 seconds of treatment. I have seen a remission on a person who got 20 sessions of CTBS to the frontal pole. That's 11 minutes of brain stimulation to remission. Kind of crazy when you think about it with the transitions. But the implications are actually huge here. So if you look at the cost of it, total room time and cost of delivery, you know, it's $2,800 just to do the O'Riordan protocol, just in terms of cost of delivery alone. 3D is $780, but not the same remission rate as Saint. Saint's not bad. It's $1,500 just to do cost of delivery. The 1D protocol is $266, not per session, per course. Right. And the abbreviated, if we could get the 1D protocol with CTBS up and running, that's $185. I told you this stuff seems to last for 4 to 6 months or so. So that breaks down to $1 to $2 a day in terms of cost of delivery. So one of the weird things about accommodating a patient's preference for a more convenient single day cost of treatment is the chair time also comes down dramatically, and the cost of delivery, you just get this for free along the ride. The cost of delivery is now almost at parity with medications. It's crazy, but you know, these brief protocols with fewer sessions and less time in the chair, no one's focused on doing trials to get the chair time down. But this is what will lead to cost parity. And remember, I don't have any randomized control trials to show you. I wish I did, but you know, once we've done that, because that's what's needed to demonstrate this. But the figure of merit, the thing to be optimized, if we're ever going to get universal access, is to work on single-day protocols that patients love and minimize chair time that payers will love. Because I think there is a pathway to get the cost of delivery down to parity with medications. And once we have that, we at least have a pathway to adoption. It's all about stripping things. These are the 3 generations of the engines that power the Starship, and you notice that they make them smaller, simpler, and cheaper every time. They don't add stuff; they take away stuff each time and make it simpler. So our priority is to optimize the sequence in various ways. Nobody likes the 30-minute intervals, right? People want to have a shift of nurses working shifts of 8 hours, so one thing to do is see if we can cut the interval down even further, shorter than 30 minutes, and we have people working on that now. No one ever thought, why is theta burst 2 seconds on and 8 seconds off? Is it because we tried all the other intervals and optimized it? Nobody has. But weirdly, just cutting, if ITBS worked just as well with 2 seconds on, 2 seconds off, or 2 seconds on, 0 seconds off, like CTBS, if that worked just as well, then for the reasons I've shown you, the economics changed dramatically. The number of patients you can run per chair changed dramatically. And lower frequencies, 1 hertz versus theta burst. If they both work equally well, 1 hertz TMS can be done on a 40-watt power supply. You can almost run it off your phone charger. It's a little bit ridiculous. Fewer pulses. We actually don't know what the minimum number of pulses required to get this plasticity effect is. And whatever we think we know is all done off of DCS. So all the things we thought we knew about every one of these has to be redone over again with plasticity enhancers on board, because the answers could turn out to be totally different. Even the intensity of what's the minimum, maximum intensity we have to go to, that drives the voltage on the TMS machine, safety and power. If we ever want to have like a little DVD player sized TMS device that runs at lower voltage and so on, then optimizing this so you could have tiny machines that would live on the wall like automated defibrillators is driven exactly by questions like this. So weirdly, the optimizations for scalability, this is all really important, interesting science, but it's different science than most of us are doing right now. And finally, do we need D-cycloserine? Anyone, I don't know if you've tried it, it's not easy to get. In India, it's really cheap, and in some countries where tuberculosis is common, you can get it for not that much. In the United States, it's about, if you order 30 tablets off the shelf, you can get it, if you can find it, $100 a capsule, right? not feasible for people doing 36 sessions a day of TMS. By the way, in 2 months, we will now have a major national compounding pharmacy will be making it for about $8 a pill. So we've solved that a little bit. But before we go all in trying to build a whole new supply of D-cycloserine, there are other molecules that also are partial agonists at the NMDA receptor. D-serine is not even, it's a nutritional supplement. It's an amino acid that is found in low quantities in the body. Josh Brown, Kerry Ressler, a bunch of the folks over at Harvard have, D-serine has been studied as a cognitive enhancer, among other things, for many decades because it hasn't been used alongside TMS before. You've even got stuff like sarcosine, which is an industrial chemical, which you can find in great quantities. It's also found in the body. It's also a partial agonist in this receptor system, and no prescription required. These things are about 30 to 50 cents a dose. So even people who are on the old-fashioned 36 sessions over 36 days could avail themselves of this if it worked. And as long as we're personalizing things, by the way, I mean, we obviously love the idea of personalizing if we have the resources to do it. But if you're in some clinic in the middle of nowhere, do we optimize it? Or because I certainly have patients who come in. We actually had a grant once to randomize people to the personalized target or not the personalized target of dorsomedial versus dorsolateral. And the first patient who came in said, well, on your wall, I saw you guys discovered this 3-minute treatment. So since it's only 3 minutes, I did drive 2 hours to get here. Could you just not put me in the study and just give me all 3? And we're like, well, actually, yes, we could totally do that. There's no reason not to do that. We don't know what it will do, but there's no reason not to do it. And interestingly, that may confer some benefits. I don't know anyone who's done all 3 until recently, but Valerie Voon out of Cambridge and her collaborators in Shanghai did do a randomized study now looking at 20 sessions over 5 days with a dual site protocol. So dorsolateral plus right orbitofrontal, or dorsolateral plus sham, or sham plus sham. What's interesting is if you look at the response rate, in orange are the single target traditional dorsolaterals, and just like us, she sees that when you do it immediately, there's not that much benefit, but you come back a week later and there's more, you come back 4 weeks later and the effect builds up gradually. But look what happens when you throw in orbitofrontal. These are the blue bars. The response is almost immediate. So they're not overall ending up better, but they get there faster. And that's also quite interesting. So I'm going to show you something that's very preliminary. I almost didn't put it in because it's very preliminary, but we are rerunning 3D or the 1D study with not D-cycloserine, but with D-serine this time. And we have also, since we're not sure what brain area is the optimal or what combination of these is the optimal, we're just doing all 3. So people are coming in and they're getting 3 minutes of dorsomedial, and then they're getting 1 minute of orbitofrontal, and then they're getting 3 minutes of dorsolateral, because you can actually do that every 30 minutes. It's not that hard to do. And giving people 1,800 pulses of stimulation is pretty standard if that's what Saint is. So, we will, or at least, but the only difference is we're spreading out those 1,800 pulses across 3 sites instead of doing all of them at dorsolateral. I can't tell you for sure what that's doing yet. I can't tell you that, but right now we're only out to week 3 with a reasonable number because we're only halfway through the series. If we were doing this lecture in 2 months, I could probably give you the full answer, but we are seeing some people say it's doing nothing for some people, but it's doing quite a lot for others. We are seeing on average 60 or 70% improvement already by week 3. And there's been quite a nice drop in the scores here. That also applies to the GAD-7. So on the GAD-7, we're also seeing these same drops. And again, if we plot all of the weekly improvement from baseline, the curve is looking, it's comparable to what we're seeing with D-cycloserine. So I don't know if we're all going to be prescribing D-cycloserine or if patients are just going to be ordering D-serine off Amazon. You can literally order it on Amazon. And by the way, the supplier that I saw on Amazon, we sent it off for an assay and we did have it tested. It does indeed contain the proper amounts of the molecule that was advertised and is uncontaminated by bacteria or heavy metals. So you can get decent D-cycloserine off Amazon. It's a little strange, but yes. All right, so we're working on optimizing this protocol. When we've got something optimal, we can go and test it. We could test it on depression. We could test it in anxiety. We could test it in OCD. We will certainly be testing on things like Alzheimer's and Parkinson's and other indications where we know TMS is useful, but it's just not practical yet. And that comes back to the question of 1 billion remissions from what? I'm going to ask, do we really need to pick a symptom? Or can we just use a more meaningful metric for actual people in their everyday lives, which is just what I want, is I just want, do I have clinically significant impairment of functioning in the activities of everyday life? Is it totally crazy to set that as your primary outcome measure? I don't think so. In fact, one of my most beloved studies of Nolan's is the Ibogaine, the MYSTIC study that came out. And there are a lot of reasons to love the MYSTIC study, but the one I love the most is the primary outcome was boldly set to be the overall level of disability on the HUDAS scale. And the symptom measures actually belong, in my opinion, where they should have been all along, which is the secondary outcomes. I'm less interested in whether it helped my depression or anxiety or PTSD, because if that doesn't translate into an overall improvement in disability level, then we're back in the situation that at least some Parkinson's patients are with deep brain stimulation. You go through this expensive procedure, your tremor gets better, but your actual functional scales don't improve that much. And so we don't just want to, it is possible to get someone's symptoms better and then not have them function all that much better, in which case you've sort of wasted your time. So here's a weird thing. Ibogaine is not something I have to pick a focal target for. With TMS, I can't just spray it. Well, we've tried to spray it everywhere, but we really probably should be this in a bit more of an empirical way. So are there neural substrates to resilience? Because that's really what we're talking about, is how do we give people resilience? And the answer is we think so. So voxel-based morphometry studies have identified areas that are thinned out in depression and OCD and so on. They've also identified areas that are thicker in people who have unusual levels of resilience. You take a bunch of people who've been through tons of childhood trauma, and there are some who never develop axis 1 disorders. They don't get PTSD, they don't get major depression, and they seem to have thicker gray matter in many of the same areas we've been zapping with TMS coils all these years. So interesting, you can also, here's a study with intracortical stimulation. In an epilepsy patient, and stimulating some cingulate areas, which led to activation of this whole network, induced what was described as a will to persevere, like pushing through emotional distress, which is a bit of a transdiagnostic thing. Some of you will have seen a lovely collaboration between the team at Stanford that originally used voxel-based morphometry studies to identify a transdiagnostic network for psychiatric illness, and teaming up with Sean Siddiqui's lesion-based data, they found good concordance of a transdiagnostic network across it. So all these areas that perseverance is elicited by stimulating, what happens if those areas are offline? Well, you don't have resilience, you don't have the will to persevere, and some form of psychiatric illness will pop up. So that's the idea. And this opens up, if it's transdiagnostic, then why are we focusing on treating any particular axis 1 disorder? If you go to the original STAR-D trial, one of the indices they put together was really interesting. It was an integrated individual burden of illness index, the IBID, for major depression, which was a kind of first principle component of how severe your depression was on the quiz, how poor your quality of life was on the QLSQ, and what your work and social functioning was. So you get this kind of this kind of component here on which you can rank someone's overall burden of illness. And what's neat is if you just look at the quiz, you can find that there are some people who are functioning much worse than you'd think for their depression scores. Their depression scores aren't that bad, but they're completely not functioning. And we all have seen, anyone who's been through a clinic and seen tons, I've seen people who can just somehow persevere and work through incredible amounts of illness. Is the sort of the Pope John Paul's types who can have terrible Parkinson's and somehow keep being Pope despite all of their Parkinson's. And then you have these other people who are just tremendously functional impaired. And if you try to pin down, well, what is it exactly? Well, they're not working, they're not getting to work, they're in these battles with their disability insurance, but it's not clear what their symptom is. And there's a poor correlation between how bad the symptom is and the amount of functional impairment. So I was pestering Sean Siddiqui for a long time to say, well, you've got all these lesion studies across all these disorders. Why don't you regress out the symptom severity and just to say, okay, well, let's just get rid of whatever expected functional impairment is there from your symptoms. Let's regress that out. Let's look at the residuals. What is the residual functional impairment, positive or negative, after you've factored out how functionally impaired you should be, expected to be based on your symptoms? Who's functioning better than expected, regardless of symptom? Who's functioning worse than expected, regardless of symptom? Because you could also do lesion mapping on those residuals, right? And a network does pop out. This is Sean and Beatrice Milano, the first author, have graciously granted permission for me to share this preprint with you. Lesion mapping based on functional disability, having regressed out the actual severity of the illness. And lo and behold, you get two lovely circuits that pop out, the dorsomedial prefrontal cortex and the orbitofrontal cortex. areas that we have spent a lot of time stimulating, in which anecdotally patients come in and say, yeah, I still have my anxiety, I'm just functioning better, I just push through it. And we just kind of write these up. We just focus on the symptom scales, but if you go off the symptom scales, people just keep talking about how much better they're functioning, even if the symptoms are there or not there. We are pivoting to this. This is starting to look like the most capable recipe. Notice there's no dorsolateral here, so it allows us to cut 3 minutes off the treatment. The other thing to love about dorsomedial is it's pretty idiot-proof. Like, you really have to be very bad at TMS to miss the dorsomedial. It's even the anatomy is also very cooperative. It's not particularly anatomically variable the way the dorsomedial targets are almost always in the same place. Frontopolar targets are almost always in the same place. They don't move that much, so you might not even need functional imaging to personalize it, because even though the personalized target may just be kind of pretty much in the same place for everybody. I'll just share with you one person. And remember, the 1D protocol doesn't work in a day, right? This is not what's supposed to happen. Unlike SAINT, where you get very fast responses, which is lovely, the 1D protocol doesn't have personalized targeting. You do not get people better in 2.3 days. It's just not something you see. But we have started pivoting to a dual target protocol using FZ ITBS for 3 minutes and FP2 CTBS for 600 minutes. And we keep the Vyvanse and the DCS4 now, because we're still figuring that bit out. We'd never seen this before. A person who goes from a Hamilton of 20 down to a Hamilton of 2 and stays there for 3 months. And she actually reported symptom improvement within 48 hours. It was very much like Valerie Boone's result, where adding on the orbitofrontal speeds things up. I woke up, this is 2 days post-treatment. I woke up ready. This is a person who's been in this current episode for more than 2 years, has been unresponsive to any med, and has essentially been in low-grade either dysthymia or depression since the age of 12. And now she's 40 years of this. And she's tried every treatment under the sun with the exception of ECT. Hadn't done ECT yet. We did this instead. I woke up ready to start doing something weird. I found that I could only lie there for about 30 minutes. Weirder. She would normally lay there in bed for 2 or 3 hours trying to summon the will to get out of bed and start moving. You see, she got bored of lying in bed after 30 minutes, got up and showered, packed, ready to leave a whole hour early. Weirdest of all. I'm curious to see if this action continues into the work week. So we're now 3 months out with this person, and she essentially is, it's almost just sad that this is she could have had this 40 years, well, not 40 years ago, but she is a different person than the one she is before. It's an N of 1, but you know, there will be others after her, and we'll see how it goes. But I do love this idea of pivoting to think of TMS uniquely, potentially not as a treatment. And I'd love to have a chat with you, and we've been chatting with Tom Insel and a few others about this. Like, do we think of this particular protocol as a treatment for indication A, indication B in the standard FDA way? Or are we saying that is the country not facing an epidemic of mental illness, but a great national deficiency of resilience? And if we flip it on its head, is it possible to conceptualize certain targets as just, we're not going to treat the disorder, we're going to provide resilience and you'll do the rest? Because that's what we're looking for, courage, not the absence. We're looking for a little more Nelson Mandela in everybody, right? To really kind of summon that fortitude to, you know, be imprisoned for 20 or 30 years and still fight the good fight and come out ahead of it and not feel defeated by what's going on. Not the absence of fear, but the triumph over it. Not the ability to, not taking away the fear, but somehow conquering the fear. That's what I'm hoping for. And because we've got this lovely group here that has spent so much time thinking about the transdiagnostic issues and how they may, I think I couldn't have a better group to think of to sit down and talk about whether we could reach those billion remissions by focusing more on building strengths than on addressing particular weaknesses. So let's end it off there. And thanks very much for your time.
Thank you so much, Professor Downar. That was amazing. I'll start by seeing if we have any in-person questions first. I know we have a ton on Zoom, but if anyone in here?
Hi, thanks for a great talk. Really exciting with the 1D study results so far. It's so striking that you don't get the results after or during the day, but in the weeks afterwards. And it's interesting also that the dual target one, it's different.
Yep.
What's your speculation of what that?
Yeah, so that's a great question. We're still trying to figure that one out. There are two explanations that come to mind that are possible. One is, I guess, more of a cellular one, and the other is more of a psychological one. So the cellular one would be that You can get the induction done in a day, but it just takes weeks for the proteins to be expressed and the dendrites to grow, and then new boutons to sprout, and spines to sprout, and all the rest of it. So it may just be that you do a bunch of stimulation, and then it takes a few weeks for all these new connections to grow. Some of you may know that there's a version of TMS that's applied to the muscles. M-Sculpt is basically a big TMS coil attached that you use to stimulate muscles and drive them harder than your nerves can. So people will go and they'll get sort of 3 or 4 sessions of intense magnetic stimulation of their abs or their glutes or whatever muscles they want to bulk up. And they don't get the effect. And obviously it takes, we're used to the idea that you can get all that done in a day, but it'll take 2 or 3 weeks for those ripped abs that you're looking for, so to speak. So, the molecular or the cellular explanation might just be that this one day thing kicks off a process, but the actual expression of that into changes in brain anatomy just takes a while. The other explanation, which is attractive, is that if you just do dorsolateral stimulation, you might not be directly treating the depression at all. I mean, you might actually be strengthening this will to persevere, this resilience, this cognitive control, this response inhibition. And so what you've done is that maybe the very next day, if you check somebody's performance on the go, no-go task, we haven't done this yet. It might be that, wow, within 24 hours, their go, no-go task is suddenly amazing. But they don't notice yet, because what has to happen is for a few weeks, they have to go down into their dark rabbit hole of rumination and then notice, oh, wow, it actually came out again. And as they keep coming out and spending less time stuck in the rumination, the depression and the anxiety and other symptoms come along for the ride as they gradually start to use this new capacity to dig their own way out. And then, of course, if you target the orbitofrontal, which is that rumination loop in the first place, then you may be short-circuited. So not only are you providing them with this cognitive control, but you're also going in and resetting this rumination loop in the first place, in which case you get a stronger effect. So I don't know if it's A or B or both. Yep. Yeah, that's a great question, because the vast parameter space of TMS or any other form of brain stimulation is there, and that's certainly the appeal of personalized targeting. So what do we do for the non-responders? We barely even started yet. We're just trying to figure out for the 90% or 99% of clinics that don't have access to personalization yet, what can we do for them before we personalize? And so I think you're absolutely on the right track that, a lot of people are within 20 or 30 millimeters of the target, but there are going to be some who aren't, and maybe that's what the 10 or 15% of. I will tell you that at least one of our dorsolateral non-responders came back again 6 months later and did the dorsomedial plus orbitofrontal and remitted. So, there may be that different targets are required, and there may be other people who will respond to dorsolateral, but they do need the personalization of a Saint type approach or something like that. So, I think we may be in a world where some people can get away without it, especially if the D-cycloserine or other plasticity enhancers are there. And other people, their brain anatomy is different enough from the default that you're going to need to go in and personalize. And what I'm really excited about is seeing what happens when we take folks who are doing Saint and are doing it on, because as I mentioned, even if you get Saint exactly right and get the target right on there, it could still be possible that plasticity will still trip you up and you won't get the response you're like, or some response, but wears off in 4 weeks or 6 weeks, and now you're back where you started. I do know some clinics that are starting to go back and take their previous Saint patients and go back to the same target with DCS in cases where they didn't get a durable response as they would have liked. And so we'll soon find out whether the best of all worlds is to use the personalized approaches of Saint or other frequency-based or other kind of personalization, but without neglecting the second piece, which is so essential, which is the neuroplasticity. I think and by the way, also for those of you who are just studying personalization and biomarkers, your error bars will be bigger if some of your patients are not doing what they should be doing because of lack of plasticity, right? So you may be on the right track with your hypothesis, and yet your signal doesn't quite reach significance because some of the people, you were in the right spot, you did everything right, but the plasticity wasn't there. So I suspect that our progress on personalization and biomarkers could potentially get a whole lot better if we remove part of the noise by giving everybody some neuroplasticity enhancement so that they'll actually do what they're supposed to do. Oh, interesting. So should it be a physiological signal or a cognitive outcome? Yeah. Well, I feel bad offering speculations when we already have somebody who is literally a recognized expert in addressing all the questions you're talking about right here, like thinking about all of the ways in which we could use these. So, what combination of cognitive biomarkers and physiological and imaging and so on. I don't know what's going to offer the best performance. I do know that depending on your clinical treatment setting, some will be more practical than others. So some places the MR scanner is right there and it's easy. Other places it might be 4 hours away and you're going to need some computerized cognitive test, or ideally a self-report questionnaire is the cheapest of all if you can pull out these folks who have response inhibition problems just by doing some pencil and paper type questionnaire. For some of them, that may be the best. So in terms of which one is the best, I think there's the best in terms of performance, but I think the key question for scalability is going to be which one is the best we can get away with in any given practice setting. So I don't know if there will be a single answer. I think we'll want lots of biomarkers of different types, and I think we want good numbers on how useful they are in terms of positive and negative predictive values, so we can figure out which ones to use in which settings.
Hello? Okay, we're going to switch to a few Zoom questions. So Diane on Zoom asks, if the patient is already taking Vyvanse, does this impact neuroplasticity with regards to the efficacy of 1D?
Oh, that's interesting. So what happens if you're already on Vyvanse? So this has come up, and typically what I've seen clinics do up until now is I just leave them on what they're already taking. So, and the grounds for that would be that if you saw the data from Andy Luchter and Scott Wilkie at UCLA suggested that people who were on those medications were doing better. So if they're already on Vyvanse, do you need to add more? Well, probably not. So.
Sense. Okay. And we'll ask one more from Zoom. Rashmi asks, what were the side effects of the 1-day RTMS treatment?
Yeah. So the question, did anyone know? Thank you for that. The one thing about a 1-day treatment is that adherence is pretty. There's no dropouts because you're just coming for a day. So the question is, did everyone make it all the way through? Everyone did make it all the way through. No one dropped out early. We haven't. And the reason for that is we just set a clear direction that it's better to run at what we are not sure is a therapeutic intensity than to have the person quit and get 0% intensity stimulation. So I will say that even with the more posterior target, the most common side effect was just the soreness of stimulation. They haven't had as many days to adapt to it. TMS pain or discomfort gets easier over time. But I would say that was the most limiting one, was that. Interestingly, for some people, you could only really get their intensity up to 60 or 70% of their MT, and many of those still remitted. So we may, so what we've been targeting when we talk about doing a simulation of 100% or 120% or whatever, it may be that we have to kind of reevaluate what a therapeutic intensity is in the context of these neuroplasticity enhancers, because that could potentially change the threshold in a favorable way.
Thank you. Do we have any more in-person questions? Okay, let's ask one.
So going back to the idea of enhancing plasticity and NMDA modulation, have you thought about using dextromethorphan, maybe with a CYP2D6 inhibitor, as a more potent
right. So yeah, so the idea of using dextromethorphan alongside, and then there are combination medications out there that use dextromethorphan in combination with other agents, which are designed to, reduce their metabolism and cause it to stick around for longer. And so the thing we may be up against there is it's not just hitting the NMDA receptor. And I don't want to present them. So for example, I'll get ketamine as an example. Ketamine is an NMDA antagonist among many other things. And so you get a rapid response in depression with ketamine, which then goes away. Whereas here, what we're doing is agonism at the NMDA receptor or partial agonism using this low dose D-cycloserine. So I don't know if it is just purely receptor. I don't know if you can draw a one-to-one mapping between the receptor and the agent and the direction and the antidepressant effect. There are almost certainly different mechanisms there. For example, in the setting of ketamine, and I don't mean to quit. I can't help but keep quoting Nolan's work because he's just done so many cool things. Blocking the opioid receptors and giving ketamine seems to abolish the antidepressant effect of the ketamine. So the NMDA piece that we all focus on for ketamine may not be purely NMDA, but it's a messy medication with lots of other effects. We also know that D-cycloserine by itself, I mean, there are papers from the 1950s where it worked as an antidepressant, but you had to give it for weeks, just like any other antidepressant. So a single dose DCS isn't expected to do a whole heck of a lot for people. And remembering these people are not taking weeks of any of these medications. It's just a 1 day. So one dose just on the day of. They're not taking Vyvanse after. They're not taking D-cycloserine after. It's just 1 day, one dose.
Hi, really interesting talk. I'm curious to know if age affects any of these treatment outcomes, and if it's the case that like younger, more malleable brains are more receptive to these treatments.
So you think we all thought that, and yet again and again, the two biggest demographic predictors of better outcome for TMS are older age. Being 80 gives you about the same chance of being 18. Like there's not really a whole like 80. Like older people do really well on TMS, and if anything, it's a positive predictor. But we don't know why, but it seems to be a positive predictor. Being female also gives you about a 1.3 odds ratio. So being older and being female gives you a better chance of responding to TMS. We don't know. At 1D, we just haven't run enough people to know what that's like. It's only open label studies. There are no randomized studies yet, and it's not even published. Remember, this is just a preprint, so all of this has just happened in the last 8 months. So it's all pretty new.
How young do these studies go? Like, have they looked in younger kids, or is this mostly like 18 to
So the
65?
series I'm showing you are just to get things rolling. The series I'm showing you all involve people between 18 to 65. Paul Corkin from Mayo Clinic is one of the recognized experts in treatment in adolescents, and we are now spinning up a case series over at Mayo to try and look at these younger populations. And so there are no theoretical risks to doing it, and in fact, one of the big holdups that many people face in treating patients. So you get lots of adolescents who the parents or they themselves would rather not take medications, but TMS over 36 days is just a non-starter during the school year. But being able to come in on a Saturday and get all the sessions at once is a complete game changer. And so as with many other populations like Alzheimer's, Parkinson's, and also the adolescent anxiety and depression group, may be the right ones. And particularly if the mechanism is not just that you're going to try and efface it with an SSRI, but if you're actually going to try and bolster the resilience, bolster the self-regulatory capacity, then we're looking at a fundamentally different mechanism that could address a lot of things beyond just, it's not antidepressant, it's not anti-anxiety, it may be pro-resilience, pro-fortitude. Like we'll need a new term for it, whatever it is.
Great, thank you.
Thanks so much. That was such a great talk and just really exciting results. So I'm an attending psychiatrist at the VA, and one of the things that we debate a lot is just what should we do as our standard for like the number of sessions per day, intersession interval, like percent MT. And so I was just curious if you could talk a little bit more about like the choice of 30 minutes in the context of like Nolan's choice of 50 minutes. With like spaced learning theory ideas, and then also kind of the increase in seizure risk as you start to get down to 20 and 10 minutes.
So there's a bunch of different factors to consider there. So one of them, interestingly, I think Nolan and I are sort of on the same page. Way back in 2017, we were having a talk in Barcelona about optimizing the intersession interval. At the time, I think both of us agreed the literature suggested you needed 60 minutes, 90 minutes. And so but then the question is that still the case when you add neuroplasticity changes, when you add DCS or when you add more dopamine or so on? So I think we just don't know. Like there's lots of animal literature on the optimal intersession interval in hippocampal slices, for example. But in therapeutic context, when you're treating people, and like I think unfortunately we're going to have to do this empirically. What I can tell you is in human embryo, they have built a model in human embryonic kidney cells. This is the Hex cell paper from some of you will have seen it last fall, where they were able to create a proxy for the same LTP and LTD process in nice, easy to culture kidney cells, which are much easier to work with than neurons. And they were able to show that you could get the intersession interval down from 30 minutes to 20 minutes, even down to 10 minutes between sessions, but 0 minutes did nothing. So there is a limit, and it's somewhere between 0 minutes and 10 minutes. We haven't tried that in humans, but we could fiddle around in animal models, but it might just be faster to do the trial. So that's an empirical question. The second one regarding seizure risk, as far as we can tell, the seizure risk of TMS may be mostly to do with staying off the motor cortex. And so if you're all that worried about seizures, one of the nicest things you can do is just make sure the intensity isn't too high. Theta burst has been shown to work just as well at 80% motor threshold as by 120% of motor threshold in the work of Paul Fitzgerald, Leo Chen, and a group out of Australia that was 100 people in each arm. And we've been seeing this as well. People are doing really well, even on 60 or 70% of the MT. So if we're all that worried about seizures, as far as I can tell, the rules are stay off the motor cortex and use lower intensity stimulation. Maybe at 7 or 8% of the MT could be fine. We took people up to 100 for this study, so that seems to be okay. But those would be, I guess, the main thing about how to do this safely. So the intervals really need to be sorted out empirically. The number of sessions to be done. I've shown you what 20 sessions on DCS is done. I've also shown you, so those people who are stuck halfway between remission and response, maybe they needed 30. Like it might just be that we could have just got 90% remission if we just brought everybody back and gave some of them more sessions. So all these things need to be worked out. And yeah, as we mentioned, a 20 or a 15 or a 10-minute interval, a lot of clinics I think clinics are already going to do that because you just getting people in for 10 hours and supporting that staff-wise is just complicated. So a 20-minute interval allows you to complete all the sessions in 6 hours and 23 minutes. And then another hour for the DCS and waiting for that to kick in fits nicely into an 8-hour shift. So if you can stay tuned for a little, the nice thing about one-day trials is, as you've seen, they're really fast. You can do a one-day trial really quickly. If you've got enough patients, you can run 30. You can run 30 of them in 2 weeks on one machine. So yes, I don't. You won't have to wait around too long for an answer to how can we get this short enough to be useful for people. I will note that even on that it's only 600 pulses times 20. So that's 12,000 pulses in a day versus Saint is safe and offers 18,000 pulses per day. This is actually only 12,000 pulses per day. So the overall dose is a little bit lower. So lots of reasons I think that we've seen it to be safe, but even for theoretical reasons, we think there's probably lots of ways to do this quite safely.
So it seems like we are fighting for Dr. Downar's time. We have over 50 people on Zoom with a bunch of questions, but I'm conscious of the fact that we are approaching noon. So I'll ask a couple of these. I think they're short, and then we'll close off, I think, with your question.
With your agreement, we could have people demand meetings.
Yeah, that sounds great. People are having, or I wonder if we could consolidate them. Would it be all right if they emailed them to somebody who would then?
Yeah, perfect.
And then I can send back a group reply.
That sounds great. All right. So for the folks on Zoom, we will get all of your questions answered and we will respond to you in a group email. So Steve asks, can you tell us again what was the targeting method in the 1D trial? Was it a variation of BMF3?
Great, thank you for clarifying that. So the first thing is it was not a personalized target. It was based on scalp heuristics, just percentages of head size and everything. We did not quite use BMF3. BMF3 was validated against Mike Fox's study that I showed you from 2012, but we decided to upgrade a little bit or update based on the causal network maps of Sean Siddiqui and colleagues with Mike Fox, who found not one, but two foci within the dorsolateral prefrontal cortex on each side. For tolerability, we used the larger and more posterior of the two, for which a scalp-based heuristic is available. We co-developed a scalp-based heuristic for that target. And so if you just look up scalp-based heuristic if you look my name and Sean's name, and you will see this paper pop up where then there's even a free online tool. So if you have a clinic and you want to go to Sean's anterior or his posterior targets, there's an Excel spreadsheet that's available on a free article, a letter to the editor in brain stimulation from 2022. And you can just it's just a BMF3 like tool. You use the same approach as BMF3, it's just different percentages.
All right. One more Zoom question, I think, and then we'll finish with Dr. Williams. What coil did you use and what was the size of the stimulated region in cc's?
Great. Oh, that's a fascinating question. I wish I knew here's what I can tell you about it. The coil we use is a forthcoming coil. It's the AMPA L coil. It's a new coil that's coming out with, and whose layout was designed by Larry Kopanen at the University of Cambridge. It's a bit bigger. I would say it's smaller than an H1 Brainsway helmet in terms of volume, like obviously, but it's larger than say a MagVenture B65 or a MagVenture B70 or a MagStim Figure 8. It's not quite flat; it's sort of spherically curved. So it's a new coil. It was just FDA. Actually, the FDA clearance on it just came a couple of weeks ago, but it's a bigger coil, and that may actually be part of it. That Alex McGear hopefully will talk on this someday to your group, but he's talked about his read on D-cycloserine creating a larger penumbra of plasticity so that you are allowed to be further off the target and hit it maybe tangentially with the field. If that's the case, then DCS should pair best with big coils like the helmet coils of Brainsway, or like the bigger your coil is, the better it should, the more likely you should be. So I don't know what would happen if you use DCS with like a smaller coil. Certainly I know a lot of people will, but our coil was kind of bigger in size than a lot of the coils that are out there, but not quite as big as a Brainsway.
Okay, thank you so much for a real tour de force, incredible scientific rigor, as well as I think all of us appreciate that you're thinking about how do you make this actually translate and scale in the real world, including the focus on disability and the non-responders, which I think really highlights that when we talk about depression in many studies, it's assumed to be about mood, whereas the impairment is really often what patients struggle with. So I think we really value that, and thank everyone for joining us, and please send us your questions. We will consolidate. I have so many things I look forward to discussing with you, including findings that Laura Hack and I have found for guanfacine, which also is another arguably a boost to plasticity in these regions. One practical question, maybe just to end off with, is by definition clinically, we're giving TMS for non-responders to the medications they're on. So do you think one ingredient is that we should actually not be continuing on what is by definition a treatment that isn't working? Meaning, is the approach you're delivering with these incredible results doing so competing against an ongoing treatment that may not be effective?
Well, that's interesting. I would say we approach that problem purely pragmatically, in the sense that when people turn up, they kind of just want to jump in the chair and do the treatment, but they don't want to go through a whole washout period of coming off the medications right away. That said, if they're getting a benefit, and if they're showing the benefit within a few weeks, I think most prescribers are going to follow the lead there, because one of the first questions patients ask is, well, the TMS is working, so when can I come off these meds? And then, there's then a discussion between the clinician and the patient about, okay, which ones are starting, which ones have the worst side effects, and sort of gradually weaning you off of them. Some of the patients in there had failed medications, but we're currently not on anything. So I think that will end up being certainly staying on. It's a lot easier to put people. It's a lot easier to put people on something new than to take it away. And this is why folks with refractory, almost like borderline patients, end up on so many different medications, because nobody follows the 1 in, 1 out rule. People just keep stacking on more of them. But it's a very good point that if we do have an effective treatment here that only needs to be done 2 or 3 times a year, that I think a lot of people will create latitude for people to get off their less effective treatments.
And lower the cost even more.
Absolutely, absolutely. Very good.
I think if we could. There we go. If we could all just thank Dr. Downar for his time and for this brilliant talk. We are so appreciative of having you here.
Thank you so much, everybody.